So we have here a diagram of our insulin receptor. The insulin receptor is located within the plasma membrane. So the insulin receptor is a transmembrane receptor located in the plasma membrane of insulin-sensitive cells. This insulin-sensitive cells refers to our adipocytes that is located in the adipose tissue, the myocytes that are located in the skeletal muscle or the heart, and the hepatocytes which are located in the liver. So it mediates the effect of insulin on specific cellular responses. Insulin receptor plays a critical role in maintaining the glucose homeostasis in the body. Insulin receptor is a transmembrane receptor formed by 4 glycoprotein subunits. So we have here our 2 alpha subunits, the one in yellow, and the one in green are the 2 beta subunits, and they are linked together by a single disulfide bond. So our insulin receptor has an extracellular part. So this is the extracellular part of our insulin receptor, represented by the 2 alpha subunits, and the intracellular part is represented by the catalytic domain. So we have here a catalytic domain, and it has also an intrinsic enzyme activity, as they have tyrosine kinase enzyme. So this part here of the beta subunit contains an enzyme, and that enzyme is known as tyrosine kinase enzyme. So because of this, they belong to the receptor tyrosine kinase part of a large receptor superfamilies called kinase-linked receptor. So because our insulin receptor contains the enzyme tyrosine kinase, so this insulin receptor is also considered as a kinase-linked receptor. So insulin receptors are not only receptors, but they can also act as an enzyme because they contain the enzyme tyrosine kinase in its molecule, particularly on its beta subunit. So the insulin receptor is considered to be a tyrosine kinase. So when we say kinase, that is an enzyme that will transfer phosphate group to a particular molecule. So in this case, the enzyme that is located within the insulin receptor is a tyrosine kinase, so that this particular enzyme will transfer phosphate group from ATP or adenosine triphosphate to tyrosine residues on intracellular target proteins. So when insulin will bind to the alpha subunit of our insulin receptor, it will cause the conformational change in the insulin receptor. So you can notice here that there is a conformational change, so that these 2 subunits here will form a single unit, and that process is known as dimerization, and it will also lead to the activation of our tyrosine kinase. So the tyrosine kinase will also be activated by the process of autophosphorylation. So when we say autophosphorylation, that is the process whereby this part here will be added with a phosphate group coming from our ATP, and that will activate our tyrosine kinase enzyme through autophosphorylation. So insulin binds to insulin receptor and induces its dimerization. When we say dimerization, that is the formation of a single molecule from the binding of insulin in this part here. So when insulin will bind here in our alpha subunit, the receptor will undergo dimerization, or it will result in the formation of a single dimer, and autophosphorylation of tyrosine residues into transmembrane beta subunit. So there is the autophosphorylation here, meaning that there is an attachment or adding of the phosphate group now coming from the ATP. That will lead to the activation of our receptor, particularly now the catalytic domain of our receptor. So we also have here another diagram whereby this shows the mechanism of action of insulin on our insulin receptors and also the activation of different molecules for to achieve its particular biologic effect. So the binding of insulin here will result in dimerization of our insulin receptor, as well as the autophosphorylation of the tyrosine residues into transmembrane subunit. So the beta subunit here will be phosphorylated, meaning that the phosphate group coming from the ATP will be attached to this part here, and this event here will lead to the activation of our insulin receptor. So this phosphorylation here will also lead to the phosphorylation of another type of protein that is located within the cytoplasm of our insulin sensitive cells, and that is the IRS. So this IRS protein here will also undergo phosphorylation. So the phosphate group coming from the ATP will also be attached to this molecule here, and this will activate also this IRS protein. So once the IRS protein will be activated, it will again also activate another type of molecule known as the PI3K. So this enzyme here, the PI3K, upon activation will also activate another, upon activation through phosphorylation, will also undergo will also recruit another type of molecule known as PDKA1, and this will also activate the PDKA1 to activate again the AKT1 or 2 molecule. So upon the phosphorylation of this molecule here, this will promote the formation of the GLUT4, as well as the translocation of this GLUT4 towards the cell membrane. So when the GLUT4 now will be translocated in the cell membrane, so as we all know, GLUT4 is an important channel. It is an important protein to facilitate the entry of glucose from the blood or from the outside of the cell towards the cell. So this translocation here of the GLUT4, due to the activation of this enzymes here or these proteins here through phosphorylation, will induce the formation of GLUT4. It will induce the translocation of this GLUT4 to the plasma membrane, and this GLUT4 here now will facilitate the entry of glucose coming from the blood towards the inside of the cell. So we also have here another important chemical compound here, the TXNIP will inhibit glucose transporter by promoting GLUT4 endocytosis. So this is the mechanism whereby insulin will act in order to promote, or in order to promote the uptake of glucose coming from the blood towards the cell. So basically in here, we have here the presence of our insulin receptors that is located in the plasma membrane. So for example, when the insulin will bind to our insulin receptor, it will activate the receptor by the process of autophosphorylation, whereby um, the phosphate groups will be attached to the catalytic subunit of our tyrosine kinase enzyme in our beta subunit. This phosphorylation here will also activate another type of protein here, the IRS1 protein. This IRS1 protein here, upon phosphorylation, will also activate another type of enzyme, the PI3K, that will also activate this type of enzyme. Then this type of protein here will activate another compound here. Then activation of this compound here will promote the formation of GLUT4, the translocation of GLUT4 from the cytoplasm towards the cell membrane. And the formation of GLUT4 is very important because it will facilitate the entry of glucose. So for example, there is of course the GLUT4 is very important to facilitate the entry of glucose, especially when there is an increased elevated blood glucose level of glucose in the blood. So GLUT4 or glucose transporter type 4 is primarily expressed in the adult tissues, such as the adipose tissue, the skeletal muscle, and the cardiac muscle. So again, there are different types of glucose transporters, and for the muscles, for the skeletal muscle, for the cardiac muscle, and for the adipose tissue, the type of glucose transporter that are located in this tissue is the GLUT4. So this GLUT4 here, this is a type of glucose transporter that is sensitive to, or that is dependent on insulin for the uptake of glucose. So meaning that they have only the ability to facilitate the uptake of glucose in the presence of our insulin. So that this GLUT4 is also known as the insulin, the functions of the GLUT4 are also known as the insulin-dependent translocation of glucose, because the GLUT4 is dependent on the presence of insulin in order for it to uptake or to import glucose from the blood towards the cell. And that cells are the adipose tissue, the skeletal muscle, and the cardiac muscle.